So we have a great lineup. Like I said, Jennifer is not here. She is even too sick to do it in Zoom. So yesterday she said, I'm going to present. I'm going to do this in Zoom. And this morning she said, no, I don't feel well enough to do it by Zoom. So we have actually a great substitute. Actually, her principal investigator, he takes off his bets because we know him. This is Hilgo Bruining. And what I can say about Jennifer is actually that she did extremely well. She is running the EEG lab in Hilgo's department. She graduated in 2006, did a postdoc in the USA. She did one in Belgium. She did somewhere else in Amsterdam at the NIN. She did a postdoc, and now she ended up in your lab.
Definitely.
How is that going?
Yeah, that's a very good catch of us. She was further trained by Eus. It's a bit of a surprise party for me this morning, but yeah, it's very sad because I was really looking forward to give Jennifer the stage because, as Arjen says, she's a very great asset for us. Just as a brief indication, she joined our team 3 years ago, and I think by this year we will actually be collecting our 1,000 EEGs in children, which is really remarkable. And she's actually, I just wanted to list some things. I put this over breakfast in her slides. I'm curious to see what other slides she has, but we'll get through them. But she's really remarkable. So in Eus van Zomeren's lab, she did overnight EEGs, and she's really one of people that can really look at EEGs and tell you what's happening there. And that's a remarkable skill. And I think something is really needed for precision medicine and psychiatry. And actually another great asset that I really wanted to highlight, and that was mainly the main reason why she would be here today, is that she's going to found something very unique, which I don't think many places in the world is actually capable of doing. We'll have a clinical neurophysiology facility for EEG investigations for psychiatry and neurology joint. So it's focused on quantitative EEG led from psychiatry, and I think that will be a great asset. So the thing that she wanted to speak about today is that she actually sees all these children herself. She has a number of students and PhDs that she does it together with, but mainly she sees many patients, so she can actually also form her opinion about the children that she sees, and that's for her a new thing. So what she sees is, that's what we actually highlight here in bigger to small terms. The more bigger, the more prevalent we see them. These are actually derived from interviews and data mining. That these children often have things like sensory sensitivity, full head, not being present, things that are not so typical for the DSM labels that we normally use. So actually we founded this NISU Neurodevelopmental Precision Center in Amsterdam 3 years ago with this primary aim to move away from dogmatic labels. For that matter, the title Undogmatic EEG Analysis. And it's not only about moving away from DSM labels, it's also a bit Cruyffiaans, I would say, to really acknowledge that in some children, a developmental disorder is a disorder of development. Not in all children, but in some children. So the question is, how do we actually sustain that claim? When can we think of a brain development that has been altered from its natural course that was predicted? And I think one of the most important elements that we use is that the brain is not born empty. The brain is born with activity that has a goal, that has a purpose, and wants to do something. And this so-called spontaneous activity, I always refer to it as an organ, has a unique property. It is probably served by a so-called balance between excitation and inhibition. So if you look at spontaneous activity, this 95% consistence of all brain activity, in fact, is seemingly random, but if you look closer and you study it properly, all these varying sizes and oscillations have a structure. And it has been shown in many experimental and animal models that deviations on different levels of investigation, that there are deviances in these excitatory inhibitory signaling or input or regulations that seem to indicate that that factor in the development of spontaneous brain activity could be a target and something of a proper handout to make this study and treatment of these children more objective. And that actually has been the focus of Klaus, my close partner in crime. He's a physicist and neurobiologist, and he's been working to make a measure to use all these variations to actually directly extract information about excitation inhibition balances from EEG. And he did that with a so-called computational model in which you can play with all kinds of variations between excitatory and inhibitory cells in silico and see whether we can actually understand how that turns out in mass brain activity. And from that, we were able to derive a method, we call it a functional excitation inhibition ratio quantification, which we also patented, and which we can use to directly estimate from a brain signal whether on average it's under the influence of more excitation, more inhibition, whether it's balanced, whether it's in certain regions. So that gives us a whole new quantified look at brain activity and whether that is actually in some way related to brain development or brain maldevelopment, and bridging it to clinical manifestations. So, most of you, I think, if you would think about EEG and how neurologists look at EEG, is looking at form. What does it look like? Qualitative assessment. And that is actually also has been the basis for the hypothesis of excitation inhibition disbalances in autism and related disorders, because we know that about 30% of these patients have epilepsy, and epilepsy can be seen qualitatively in the EEG. So to actually validate our method, we thought, okay, we need to bridge these hypotheses on qualitative EEG abnormalities, which are about present in half of the patients with autism, to the quantitative one. And indeed, what we found is that children with and without visual EEG abnormalities like spike waves, epileptic form change, background abnormalities have profoundly different EI balance regulation or dynamics than the ones without. So that was the beginning of, I think, a success story to create a breakthrough for objective measurements in these developmental disorders. And just to give you an example, in these 12 minutes, one of our most profound examples, we've been working for quite a while on a new treatment. It's a repurposing treatment using a diuretic drug which influences the properties of GABAergic inhibition, that we could actually really clearly show that this excitation inhibition quantification directly from EEG could be used to track effects of this treatment and actually test hypotheses on its supposed actions and mechanisms. So what you see here is that we have children treated with this treatment, bumetanide in the red and blue with black with placebo, and the standard measurements of power in the EEG, the central frequency, and here this novel EI ratio were only influenced in the treatment group and not in placebo. And this might seem trivial for other fields of medicine, but in neurodevelopmental disorders, this was really the first time ever to actually take EEG along in a randomized controlled trial and to really objectively show in a placebo-controlled manner that we could track these kind of hypothesized mechanisms in actual brains of children with a new mechanism-based treatment. And another example is that we are together with a lot of other more clinical groups, like those of Matthijs Verhagen, and to make this translational cascade from cellular EI ratios to real brain EI ratios, that we focus therefore on certain genetic syndromes. You probably know there's many genetic disorders in neurodevelopmental disorders, and this is one. It's called STXBP1, a gene that does something in physical docking in the synapse. And here we can show purpose of the method, that you see that these children are really affected by have profound lower FEI, so they have very dominated inhibition. And also here you see that they also have a lot of excess slow activity. So to put it simple, these children have very slowed, inhibited brains. They also have a lot of seizures. About 70% of these children have epilepsy, and everybody would think, okay, seizures means dominant excitation. But here we see that if we measure on the mass brain activity how that actually turns out after all kinds of homeostatic interactions take place, the counteract is true. And this is very crucial because we give these children drugs that even enforce inhibition further. So this creates all kinds of new hypotheses that we, together with iPSC models and all kinds of other intermediate steps, try to link together to really understand how cellular events actually cascade into these new measurements on the more clinical level. So for today, obviously, the topic is individuality and precision science. And just to give you a flavor how Jeanne said it's really important to bring this to the bedside, we actually have now nearly completed the first multiple N-of-1 study in which we take along all these previous notions on effects and EEG prediction models and to improve our clinical endpoints, because that first slide is crucial. You have to test the things that you see in the speaking room and don't turn back to other dogmatic instruments. And we have now included 115 of these children that go through 9 consecutive EEG measurements, 36 personalized, patient report outcome measurements, and a lot of other measurements to really provide detail what's happening in between and to really track the dynamics where patients are their own control. And this will be a whole new body of data and science that we can really get into the new field of studying individuality, but also, which is very crucial, to acknowledge individual complaints and linking them to individual hypotheses of mechanisms and manifestations. And this is how that looks like. So every Tuesday, on a one-to-one basis, we see these patients, we link the prompts, the quantitative EEGs, the questionnaires in a sort of profile that is now being further developed into a genuine sort of company and product. But it really gives us a possibility to integrate all these levels of thinking around the individuality. But the cornerstone is this quantification of EEG. And this is how the flat maps look like. If you have a new pipeline, how we extract these EEGs, we can immediately look at different regions, and all these colors indicate different, either individual differences or group level differences that we can immediately use to test our hypotheses either for science or clinical decision making. So individual is key. Giannina Christian will later on present a very nice story slam on how we really do it on a more profound modeling way to integrate supervised and unsupervised methods, to integrate all these clinical features around the individual from EEG, cognition, etc. And Aditya Sharma has a very nice poster if you want to get some more flavor of how that technically works, this EI method. But at all, I think this is a great effort where all these people in NSU integrate around these patients to make this picture really true and bring it to the promise of the parents and their individual desires. So the conclusions for Jennifer would have been that this QEEG data has really potential to bridge important labs not only in translational science but also between disciplines, because this KNF facility will be based in a new center for personalized medicine in the Emma Children's Hospital, where we interact obviously with pediatric metabolic diseases, and I think that will be crucial to make these links and put actually psychiatry at the center point of that. Thank you very much. How was that for time?
You're perfect on time. Thank you very much, and thanks to Jennifer for her work.
Yeah.
So you have a great team. How many people are there in your team?
So yeah, there's a clinical team, a core clinical team. That's about 15 people, students, residents, PhD students, Jennifer leading the EEG, and psychologists, and a very dedicated clinical coordinator, which we could lure away from the Anthony van Leeuwenhoek. Yeah. And then around it, there's the spheres of the lab of Matthijs, from Klaus, and in Nijmegen. So there's a whole different islands that really interact. But the funny thing is, even the cellular people know about the individual patients that they're studying. And I think that's really inspirational, right, for everyone. So you built on this translational team. So this is Klaus, actually. He's probably here in the room. Matthijs and Ruud. I see many familiar faces. So you have a translational team. So actually,
what made Jennifer join your lab? Because he was in a great lab already with us, and so how did that work out? How did you convince to join your team?
She was looking for a little bit more daylight, I think, because she was always sitting in the dark during the night time EEG. So.
Oh, you hear it. So you don't leave your coworkers in the dark.
No, but you know, change of atmosphere, new kitchen. She was really happy to work with us, but that has been going on for long. And one thing that she really liked, and I think that's a great opportunity of NSU, is the actual interaction with the patients themselves and to test individual hypotheses. That was something where her former expertise and the new setup could really facilitate one another, synergize. Yeah.
Okay, so I don't see any Slido questions. Is there anyone who has a question for Hilgo at this point? Because there are no Slido questions at this point, right? I see no. Okay, so you start using Slido if you want to ask questions to the reporter. Maybe it's good to go to the next presentation. Thank you very much for your last call here. Thank you very much. Okay, great. Well, let's go to. Can you hear me?
Let's go to a very different topic. It's the topic of consciousness. And I think all of us being just around or neuroscience or in neuroscience, you have at some moment been thinking about consciousness, I think. And I think several of you have been coming in contact directly with consciousness because you went maybe into surgery and then you lost consciousness, you know. And it has always been quite enigmatic that these compounds that are used in surgery and that bring you to unconsciousness work in a split of a second, you know, and people have always been thinking about how this works mechanistically in the brain. And it's a real pleasure today to have Mototaka Suzuki talking about this topic because he discovered basically the fundamental mechanisms by which this switch in consciousness works. You know, so he looked at cortical layer 5 neurons in humans, and he found out, in fact, that there are drastic changes that can occur when you apply anesthetics to these cells and the circuitry, in the disconnection of, let's say, the dendritic arbor of these cells and the cell body. There was a discoupling of activity. He will tell you about that. That work has been published also in 2020. He has a beautiful Cell paper, so if you want to read about it in full extent, go there. It's a great paper. I also was really amazed by the fact that he also developed all kinds of beautiful tools to do this work. You know, so for instance, he developed small kinds of microscopes by which you can send light into the brain, but you can also measure with the light into the brain, so you can stimulate and measure at the same time. He calls this a micro periscope. I saw Mototaka. And so this is a real important development because that allowed him, of course, to do his studies. So without further ado, Mototaka, I would like to invite you, not forgetting to say that you're assistant professor at the University of Amsterdam, and that's where your group is at the moment. Please tell us all about your research of the recent years. Okay.
Thank you very much for the great introduction, and it's my great honor to be here to present my recent finding related to this. This question: how does general anesthesia suppress our consciousness? Can you hear me, by the way? If I speak in the microphone. Yeah. Okay. Okay. Thanks. Oh, thanks. Okay, so I just directly go to the mystery. So many years ago, I learned the mystery of general anesthesia. In the awake brain, as you know, the many brain areas communicate with each other through feed-forward and feedback pathways. But very, very interestingly, during anesthesia, for some reason, the feedback signals, red arrows, are selectively suppressed during anesthesia. And the mechanism of why this happens and for such selective disruption of feedback signaling is unknown. So that mystery really fascinated me. And importantly, the general anesthetics, does not simply suppress the entire brain. Let me give you one example. This is the very, very famous discovery of orientation-selective neurons in primary visual cortex, and which was the Nobel Prize winning the study. And in this, I'm still not used to it. So this particular example shows that the recorded neuron is responding selectively to the vertically oriented bars. And as you know, the discovery was made in the anesthetized the cat brain. So the feed-forward signaling and the basic neuronal functions is intact. They're relatively unchanged even during general anesthesia. So for some reason, this selective, magical suppression of suppression of feedback signaling is happening, and we don't know the mechanism. And so among many, many possible targets, we hypothesize that the layer 5 pyramidal neurons are involved in this process. This is because these cells are the major driver of cortical and the thalamocortical loops. And secondly, the feedback signals are known to reach this distal end, the end of the distal dendrites, while the feed-forward signaling is coming to the perisomatic region in layer 5. And as I showed you in the Huber and Wiesel the example, this pathway, feed-forward pathway, is functional even when the animal is anesthetized. So we hypothesize that these cells are involved in the process. So we ask the question: if we are able to keep this feedback signal relatively unchanged while changing the brain state only, does the brain state affect the propagation of feedback signals? The anesthesia might affect the local dendritic. The anesthesia might affect the local dendritic excitability here, or maybe affecting the other unknown mechanism. So to answer this question, we took a novel approach combining optogenetics and electrophysiology and a novel micro-optical probe. I don't think that I need to explain this to this audience, but the last part, the novel micro-optical probe, is what I developed in 2017, which is combining this tiny the prism, which is 180 micron by 180 micron with a green lens and the optical fiber. Because of this lens, the light doesn't propagate, but become very focal, so that the stimulation or imaging can be really focal. And the tool allows me to stimulate a very specific the neural tissue, which is about 0.1 millimeter in the mouse brain, which is the distal end of the dendrite. And combining all these three techniques, now we are able to express the optogenetic actuator, channel rhodopsin, only in the layer 5 pyramidal neurons, and then the micro-optical tool allows me to stimulate just the top of the dendrite, which is the 0.1 millimeter, and the electrical recording, the depth-specific electrical recording allows me to how this dendritic depolarization propagates toward the cell body while I'm changing the brain state, from anesthesia to awake, or vice versa. So that's the technique we use. And as I showed you earlier, the mystery was that why the feedback signal is selectively disrupted by general anesthesia. And we found that layer 5 pyramidal neurons have a very, very interesting property to explain this. That is that the feedback signals, in this case simulated by the optogenetic stimulation with this micro-optical tool, has evoked equally strong depolarization at the distal dendritic region in both states. But in the awake state, this depolarization rapidly and strongly propagate towards the cell body and evoke action potentials. But during anesthesia, this strong depolarization at the dendritic region is strongly suppressed along the dendrites and evoke no or few action potentials, if any. And furthermore, we found that the selective, the disruption of signals is strongly affected by the metabotropic glutamate receptors and the muscarinic acetylcholine receptors. And interestingly, it is known that this higher order thalamic nucleus is activating the metabotropic glutamate receptors, and these receptors are not activated by cortical activations. So it means that this higher order thalamus was found to remotely affect the cortical coupling between this distal dendritic region and cell bodies. So the next question I'm trying to answer is that first, which type of metabotropic glutamate receptor, there are 8 subtypes, are actually doing the job. So is doing affecting the coupling of cortical pyramidal neurons. So, and it is also known that group 1 metabotropic glutamate receptors activate the intracellular calcium release from endoplasmic reticulums. So this downstream effect might be the reason of why I see such a big differences between the anesthetized versus awake states. The second question I'm trying to answer is that how neuromodulators, including the acetylcholine, noradrenaline, the dopamine, and serotonin, these neuromodulators and higher order thalamic regions, as a whole, contribute to anesthetic-induced unconsciousness and emergence from anesthesia. I want to have a clear picture, and here again, my hypothesis is that the metabotropic glutamate receptors are the key target for the mechanisms. And the third question, how about sleep? Does sleep also decouple cortical pyramidal neurons like general anesthesia does? So as you know, that there are 2 types, the REM versus non-REM sleeps, and there are the important differences. And as you know, that we typically dream during REM sleep, and the REM sleep has some similarities with awake states. So are the cells decoupled during REM sleep or not? So there are any differences between these 2 sleep states. And as you know, the sleep has some interaction with general anesthesia, so this is not unrelated topic. And the last question is that do metabotropic glutamate receptors and muscarinic acetylcholine receptors, these two receptors are shown to be important for the coupling between these 2 compartments. And indeed, my preliminary studies show that the 2 receptors interact intracellularly. So this might be another reason why I see such a big difference between the 2 brain states. And through answering these questions, we better understand the mechanism or cellular molecular mechanism of general anesthesia, and that I believe to contribute to development of safer, more effective anesthetics with less side effects. And the theme of this year's the meeting is the individuality, and indeed the effect of general anesthesia really depends on the person with different medical history, and needless to mention heart disease. But I also know the cases where the anesthesia has some very bad post-surgery effect on the patient with dementia, including Alzheimer's disease. So there are interactions between the general anesthesia and the neurological diseases. So we do need to better understand the molecular cellular level mechanism of general anesthesia and its interaction with diseases. So for the individually tailored treatment and anesthesia. So that's it. Thank you very much for your attention. I take this.
Okay, Mototaka, thank you very much. Looking at the audience, questions? Are there questions for Mototaka? Maybe I can ask you the first one, Mototaka. There are, of course, natural inputs to these cells. I guess, natural inputs normally coming in at T cells. So is the cholinergic input, can you test the cholinergic input as a natural input, for instance?
Oh, sorry.
Can you test the cholinergic input to these cells as a natural input to these cells?
Yeah.
So how do you think that is normally affecting the processing of the layer 5 pyramidal cells?
So simply speaking, the acetylcholine level increases when we human or animals are awake.
Right.
So
because cholinergic levels change during sleep and wake, so they may also affect the.
Yeah.
Okay. All right.
There's 3 questions over here,
Good.
Chris, on the Slido.
All right. Let's read them. So, how do you guide the process of layer 5 neuron stimulation? How can this be so specific? And how do you know that you're indeed stimulating only this layer?
Go to the microphone, Mototaka. Yeah.
So the specificity, question about the specificity of the targeting.
Microphone.
Sorry. Let me understand how the process of layer.
So are you not targeting other neurons?
So how do you get so close? How do you get so close to layer 5 with your technique?
Well, first of all, I skipped the detailed information, but I use the genetic approach to target only to express the channel opsin optogenetic activator only in these pyramidal neurons, in layer 5 pyramidal neurons. So it's cell type specific.
Okay.
And then the tiny micro-optical tool allows me to focally stimulate
so it's the optogenetic signal combined with your periscope that actually makes the cell specificity. So what? All right. So we go to what is AIC? What is the role of AIC in changing? I have no idea what AIC is.
No.
Let's go to the second question. What sort of experiments are you planning in order to perform to find out what metabotropic glutamate receptors are involved in coupling? So what is your next step, basically?
So the simple experiment is that there are more pharmacological agents which target the specific subtype of metabotropic glutamate receptor. So that's the first step.
All right.
So Mototaka is normally the glutamate that activates these metabotropic receptors coming from spillover of other inputs, or so how does it get there, basically?
Yeah, so that's an important point, and not just the glutamate, but there are some conditions where the metabotropic glutamate receptors are activated.
So can you speculate when these conditions start to arise?
And it also depends on the cell types and also the layer. So it all depends on which cells receive that. So for example, in the case of layer 5 pyramidal neurons, and especially layer 5B cells, are activated only by the higher order terms. So that's very, very important. So no cortical inputs can activate.
Okay. Right. So maybe is there time for another question, Arjen?
I think very little, like 15 seconds.
Okay. maybe try the second one. So these are, of course, interesting findings. This is the question of the second question. But do you want to look at the role of other cell types in other layers? So basically, is layer 5 everything, right? That's the question.
So in the paper, of course, I got kind of criticism, and so I also studied the other cells, other cell types, and only those cells have that kind of very clear differences.
Okay, much work to do still, Mototaka.
Yes.
Okay, give him a big hand.
Thank you.
Okay, thank you.
All right. So who's next? Next is Betty Tijms. I know Betty already for 10 years. She graduated about 10 years ago, I guess, in Edinburgh. But so when I looked at her CV, she's actually a psychologist. So that is not so funny. But on top of that, she is a bioinformaticist. So she has 2 master's degrees. She did her PhD in Edinburgh on Neuro-Informatics. I saw her working with MRI signals. I saw her working with EEG. She's now onto all the dimensions that actually need to be inspected in the field of Alzheimer's research. She was awarded with a VD grant very recently. and she's appointed as associate professor in the Alzheimer Center Amsterdam. This is Betty Tijms.
Yeah, thank you. So maybe some of you are wondering, how do you go from psychology to neuroinformatics? Well, I started studying psychology because I'm really interested in how we differ from each other. Why do some people respond really badly to damage in the brain and other people are more resilient? But psychologists, they treat the brain a bit like a black box. And I moved into neuro-informatics, where you combine neuroscience and informatics. And in that field, I was able to learn how to develop methods to really study the individual as well. And today I'm going to share with you a bit on how I apply those methods in Alzheimer's disease. So Alzheimer's disease is the most common cause of dementia. It is pathologically defined by amyloid plaques and tau tangles in the brain. But how these plaques end up there and how the brain responds to it, that's largely unknown. We do know from tissue proteomics and RNA-seq studies that many processes are involved. But these studies, they also treat all patients as being part of one group. But people differ a lot from each other. So an important question is how to study this in patients and how to apply that to individual people. Well, this is where cerebrospinal fluid comes in, and I will call this CSF from now on. So you can obtain CSF with a simple clinical routine procedure, a lumbar puncture. And in the CSF, there's proteins. And amyloid and tau can be measured in the CSF, and many studies have shown that these are robust biomarkers for Alzheimer's disease. But also in these markers, we see differences between people. So the first studies looked into subtyping based on just amyloid and tau, or maybe also ubiquitin, and found evidence that at least 3 subtypes in Alzheimer's disease exist that show different combinations of these proteins in terms of their levels. But this is just a little set of proteins. There's many more proteins that can be detected in the cerebrospinal fluid. And we took advantage of such proteomics to identify whether more subtypes or more biological processes associated with those subtypes could be discovered. So this is something we did 2 years ago, and we discovered 3 subtypes within Alzheimer's disease that showed very distinct biological processes. One subtype showed hyperplasticity, increased tau levels, and increased amyloid metabolism. Another subtype showed evidence for innate immune activation, and a third subtype showed indications of blood-brain barrier dysfunction. And this has potential therapeutical implications. So should we treat all these patients similarly? I don't think so. For example, if we look at BACE, which is an indication of amyloid metabolism, we saw that the levels were highest in the neuronal hyperplasticity subgroup, but they were lower than controls in the other 2 subtypes. So you can imagine that a BACE inhibitor might only work for the first subtype and might be detrimental for the other subtypes. But these results were just from 2 cohorts. And replicating something in 2 cohorts is nice, but it's not good enough. You need to replicate it in as much independent data that you can put your hands on. And also, even though we measured more proteins than 3, new techniques allow to measure even more proteins. So this gives exciting opportunities to maybe discover more biological processes and maybe even more subtypes. So what I can show you today is still work in progress, but it's new data from more than 600 individuals from the Amsterdam dementia cohort. And we identified now more than 3,000 proteins, which was more than double than we previously identified. And about a third of these proteins were associated with Alzheimer's disease. And this is what we use for clustering. So clustering is a data-driven technique. And I used non-negative matrix factorization, which is one of my favorite algorithms, because it can cluster both the proteins and the patients at the same time. And now we found 5 subtypes. And these are the results. And I'll step through this heat map in a bit more detail in a second. So in this heat map, you see columns and you see rows. And every row reflects the protein concentrations of one protein. And in every column, you see the average concentration for a particular subtype. The levels are scaled according to controls, so the red colors mean that that subtype has on average higher levels than controls, and blue is lower. So the first subtype was really similar to the first subtype that we previously observed. It showed indications of neuronal hyperplasticity, high amyloid metabolism, and also high tau levels. Another subtype was also closely similar to a previous subtype we identified with innate immune activation. And now that we measured more proteins, we were able to identify that these individuals seem to also have a dysfunction of their choroid plexus. They also showed low tau levels. And then the third subtype showed low concentrations of proteins associated with plasticity, but high concentrations of proteins that are indicative of blood-brain barrier dysfunction. These individuals also show low tau levels. Then there's a new subtype that also showed indication of hyperplasticity in combination with innate immune activation and indications of dysfunctional autophagy. And then the last subtype, which was also new, showed indications of retromer dysfunction and disrupted proteostasis. And the first 3 subtypes all show high tau levels. So what you do when you find these subtypes, you need to characterize them. So one of the things that we looked into was to compare them on proteins that were not used for clustering, like base 1. And this was increased in the first 3 subtypes only and decreased in the individuals who have brain barrier dysfunction. And we also looked into the genetics. So ApoE4 is a common genetic risk factor for Alzheimer's disease. And what we saw was that individuals with blood-brain barrier dysfunction had a slightly higher proportion of E4 carriers. And the E4 allele has been associated with damage of the blood-brain barrier previously. What is also interesting is that when we compare the individuals based on Alzheimer's genetic risk and protective factors that have been recently discovered, we saw in the heat map on the left, I think it's right, that the different subtypes showed involvement of very different risk factors. So this suggests that these biological processes that we pick up in the CSF might reflect also the differences in the Alzheimer's genetic architecture between individuals. We are also comparing them on a bunch of different clinical markers, but we don't have time to go into everything. But what you can see here is a hazard plot where you can see that people at a pre-stage of Alzheimer's disease show differences in decline. So the top line is controls, and these are people with normal cognition and normal Alzheimer's biomarkers, and they hardly progress to dementia within a period of 5 years. And all the people with MCI and abnormal amyloid, they do. And if you have the subtype with both hyperplasticity and innate immune activation, then the progression is even faster. So it has clinical implications as well. So that brings me to the end of this update of my research, which is still in progress. And what I've shown you today is that, well, CSF proteomics is really cool and you should really look into it. We find robust subtypes now in a third independent cohort. And this is really special because all these cohorts, they used different techniques to measure the proteins. So if you have converging evidence for similar processes using different techniques, that's a very robust finding. A previous study looking into subtyping with tissue proteomics also found evidence for similar subtypes. And we show that if you increase sample sizes and the number of proteins measured, that you can further refine these subtype definitions. And this is very important for therapies as well. For example, 3 subtypes had similar tau levels, but very distinct biological processes involved. So you need to have more proteins to kind of get into what is going wrong in which individual patient and tailor the therapies accordingly. Thank you.
All right. Thank you very much, Betty. So your team is somewhere in this large group of people. How many team members do you have now on your VD grant project and other grants?
Well, yeah, so it transitions a bit. There's 2 postdocs working in my group and about 7 PhD students.
All right, so it's
yeah.
a sizable team. Is that the ideal dimension?
But this is also being, some of these people are being part of different projects as well. And
yes.
That's really, we're a big team together.
Right, right. I see 2 hands here in the audience. Okay, let's first Hilgo and Narik.
Yeah, great talk. Obviously very brave that you could replicate so far in so many different samples, but I was triggered by your comment about the underlying heterogeneity in other features. So how do you know that this is actually disorder specific? Would you not be interested to test these profiles? For instance, the blood-brain barrier leakage or the neuronal hyperplasticity is definitely not specific for Alzheimer's. So it's not only a question, but also maybe an invitation. Would it be interesting to test this in other...
Yes.
Yeah.
Yes, in other patients, for sure. That's not specific for Alzheimer's only. It's really relevant for other diseases. And the choroid plexus, for example, plays an important part in multiple sclerosis, for example, where you can also see inflammation, or, well, can see there is inflammation in the brain. And what I would find even more interesting, and maybe there are students in the room who get excited by this idea as well, is to translate these clinical findings back to the bench and see what the mechanisms are that can explain these differences between people.
I think I would also start with a very young age, don't you think? Developmental disorders, and there the toxicity over the blood-brain barrier. autism being the same as Alzheimer, that would be a provocative thought, I think.
Yeah, I'm unsure. It would be nice also in younger people if you could obtain cerebrospinal fluid. I'm not sure if it could happen, but that would also be really insightful.
With the right question, everything is possible.
All right. Can you give the microphone to Rick? Rick, please.
Thank you. Betty, I was wondering, how do you differentiate between subtypes and disease states and stages? And maybe the follow-up question, have you profiled the same people after follow-up times, and do you see that these subtypes stay stable, or can they change from one subtype into the other subtype?
Yeah, and let's combine that question with the second question on the board. Are other proteins involved, actually? You now clustered it to Alzheimer's and tau, and you find 5 subtypes when you look at the other genetics. So disease states, subtypes, proteins.
Okay. Yeah, so the clustering itself doesn't, so amyloid tau were not part of the clustering, but they're outcome measures to compare it on. And all the definitions are based on proteomics only. For disease stages, so our patients are across the clinical spectrum, and already in the preclinical stage, we see these really pronounced subtype protein expression levels. We also have work where we find 2 subtypes in cognitively unimpaired individuals with normal Alzheimer's markers, albeit a bit less pronounced. And these individuals only showed changes over time in amyloid and tau. It became more abnormal. So to me, that is an indication that these profiles are being driven a lot by genetics as well, and maybe they can be upstream even of the amyloid plaques that we detect in our biomarkers.
All right. Thank you very much. I think we need to stop here. Thank you for starting right. This is Betty Tijms.
Okay, thanks, Betty. Let's go to the next speaker. That will be Jeanne Savage. So she will be talking about real individual differences, and let's make the connection to Betty again. She said, you know, there may be the molecules that we are looking at maybe even have a background in genetics, and that's where Jeanne will take us. She will be talking about the individual differences that exist in the population. Take you, take me, take the group. Basically, you will see that there are different vulnerabilities to develop disease. Betty, Jeanne, you're looking in particular, and today with us, you're taking us in the direction of addiction research and alcohol misuse and the genetics and the environmental factors, I think, that contribute. So we're going to learn a lot about that, I think, right? Okay, the floor is yours. Jeanne.
Right. Thank you, and thank you so much for having me here today. I do feel a little out of place following up all these great neuroscience talks because I'm not a neuroscientist at all. I'm a statistical geneticist at best. So I hope I can still share something that's of interest to everyone in this audience today and everyone following online, even if it's not directly neuroscience research. So my research is interested in looking at the genetic etiology of alcohol misuse primarily. And in this research, I'm trying to understand how we go from DNA, this very micro level, basic unit of biology, all the way to the other side of this macro level complex behavior like alcohol misuse. And even though I'm not a neuroscientist, I know that there are things like brains that come in between. And there are also a large number of different biological, psychological, and social factors that influence these in-between pathways. However, in statistical genetics, we typically just look at these 2 endpoints of the spectrum, the DNA and the behavioral phenotype, with not a lot of attention paid to the in-between bits. So a typical method that we use is called a genome-wide association study, or GWAS, where we take we take millions of individual locations across the genome, shown by chromosome here on the x-axis, and we look to see if there's differences in the allele frequencies between, for example, cases and controls of a certain disorder, with the significance of association shown on the y-axis and the negative log p-value, so higher values mean stronger associations, and then the red line being the threshold for significance after multiple testing correction for all of these different locations. So in an early GWAS of alcohol use disorder, with just a few hundred cases and controls, there was a significant association demonstrated in locations of the genome that contain genes that code for alcohol metabolism proteins, such as ADH and ALDH. And these are very robust associations, but they only explain a small portion of the heritability of alcohol use disorder and alcohol misuse. So the direction of the field was, or the idea that took hold in the field was that there are many more genes involved, but they have small effects, so we need to have larger samples in order to detect these smaller effects. So fast forward a few years, and we've gone from a few hundred cases and controls to over 50,000 samples. And yet the results still look very much the same. So there's still just one robust association with alcohol use disorder, which is again in the ADH alcohol metabolism gene. But what I think this approach misses is a lot of information that we have from neuroscience, from psychology, epidemiology, and other fields about the in-between bit. So we know a lot about the etiology of alcohol misuse, and we know, for example, that when we talk about alcohol misuse, we're actually talking about many different dimensions, many different types of alcohol use behaviors that can be problematic, which themselves have many subcomponents and many different constellations of symptoms or problems that can differ between individuals. And we also know that these domains have different risk factors, have different associations with, for example, other types of psychopathology. So the nuances between these dimensions and between individuals seem to be important to take into account. So that's what my research now is trying to do, look at individual differences in alcohol misuse from a few different approaches. For one, looking at subgroups of individuals that might have more similar patterns of alcohol misuse and therefore might have a more similar genetic etiology. Considering the distinctions, possibly genetic distinctions between dimensions and types of alcohol misuse. And looking at how the environment can modify genetic risk for alcohol misuse. So I'll just give a taste of a very quick high-level overview of some of these different types of research. So in the first vein of research, I'm looking at subgroups of individuals. I carried out a latent class analysis in a sample called Spit for Science, which is a large, deeply assessed sample of college students in the US. And here we found that there were 3 different subtypes associated with alcohol misuse with different alcohol problem profiles. One low-risk group, shown in red. One group, shown in green, with high levels of internalizing problems like anxiety and depression, and high levels of alcohol use disorder symptoms, but not high levels of consumption. Then a third group, shown in blue, with high levels of externalizing problems like impulsivity and antisocial behavior, and extremely high levels of both alcohol use disorder symptoms and heavy consumption. In a more recent follow-up to this, carried out by my master's intern, Excitation, we conducted a follow-up to this using a similar method in a larger sample of adults from the UK Biobank. And here we found a somewhat different pattern of associations, but we did see that there was again an internalizing group that contained a lot of former drinkers and lifelong abstainers, as well as 2 groups engaged in similarly high levels of alcohol consumption, but that differed in whether or not they had associated clinical problems like an alcohol use disorder diagnosis or a variety of other internalizing and externalizing disorders. And in this larger sample, we were able to compare differences in the genetics between these subgroups, again using a GWAS approach. And here we saw again that the strongest contributor to group differences came from the alcohol metabolism gene location, but we were also able to identify a large number of novel loci that were not previously associated with these more unidimensional measures of alcohol consumption or alcohol use disorder, and also were not associated with the other psychiatric disorders that were used to define these groups. In another type of study, in collaboration with some of my colleagues from the US, we carried out a genetic investigation of different dimensions of alcohol misuse by doing a GWAS of each individual item from the audit alcohol screening questionnaire. And then we used the genetic correlations between these items to see which items were genetically similar or distinct from each other. And this was used in a latent factor approach, so we were able to identify 2 latent factors, latent genetic factors underlying these different items. one that was associated with the alcohol consumption items, and another associated with the alcohol problems items, and these factors were genetically distinct from each other. And interestingly, these two factors, these consumption and problems latent factors, had similar patterns of genetic correlations with some domains, such as other substance use and impulsivity dimensions, but they had opposite or different patterns of genetic correlations with other health-related domains, such as psychopathology. So alcohol problems was genetically correlated with other measures of psychopathology, but consumption was not. And we also found that the genetic influences specific to drinking frequency were correlated with high levels of socioeconomic status and were in general protective of other physical and mental health outcomes. So the distinctions between these dimensions seem to have important implications for the genetic etiology, but may also have some important clinical implications as well. And in a more recent follow-up to this, I've extended this same type of analysis using additional alcohol-related items from UK Biobank. And so I found a replication of the problems factor and the consumption factor with similar genetic correlations as in the previous project, but I also found an additional novel factor that was not previously identified, and this was associated with higher levels of specifically beer drinking, but not other types of alcohol. It was also associated with a higher likelihood of receiving advice from a physician about reducing one's alcohol use, and an actual self-reported decrease in recent drinking. So a bit speculative, but this might suggest that this is capturing some genetic influences on responsivity to intervention. I don't know for sure the causal direction there, but I think it's a really intriguing area for follow-up. And finally, something that's often neglected by genetics researchers, but is really, truly, extremely important for any kind of behavioral outcome, is the environment. So one way that we can look at how the environment influences behavioral outcomes is to use a statistical interaction in a GWAS-type approach and test at each location in the genome whether there's an interaction between genes and environment. So one example that I've done here is looking at how drinking habits interact with socioeconomic status, or rather how genes interact with socioeconomic status to predict drinking habits. So as we saw from the previous few studies, socioeconomic status is an important confounder or important influence on consumption and drinking behaviors. And so in this particular study, we found that there were two loci that had a significant interaction between socioeconomic status and the genomic location, the variants in the genome, to predict individual differences in drinking habits. And these were loci that did not have a direct main effect on drinking habits, that they were only important for the interaction term. And in addition to looking at these individual loci interactions with the environment, we can also use a more aggregate approach called polygenic scoring to create an individual level score of genetic risk by basically taking a weighted sum of each person's risk alleles and protective alleles across the genome. And that gives an index of each person's overall genetic risk for alcohol misuse outcomes. And in some work from my former colleagues, we've seen that these types of polygenic scores are moderated by the environment. So for individuals that have a higher genetic predisposition for alcohol misuse, they have that expression, or that predisposition is expressed more highly in environmental contexts that are more permissive towards alcohol misuse, like for adolescents having highly deviant peers. And also that these polygenic risk scores, the relationship between genetic risk and alcohol use problems is mediated through a number of factors, including personality traits. So this actually gives some insight into the mechanisms that link genes to behavior. And so these are the types of approaches that I want to link to the other analyses that I've just shown, where we're finding genetic influences on subgroups and dimensions. And I want to take those influences into these types of models to better understand the underlying causal etiology. So those are my next steps in research. So just to conclude, I hope that this has given a little bit of an insight into how we can improve efficacy of gene finding in particular, but more broadly on studies of individual differences by leveraging the knowledge that we have from multiple fields of study, by taking multiple different methods with complementary strengths and weaknesses. Although I don't want to be over-promising and saying that this is a magic wand, there's still a lot of complexity in these results. It's going to require a lot of work and a lot of collaboration between geneticists and neurobiologists to make sense of these genetic associations and actually bring them into clinical practice. And I hope in this brief time I've also given you a little bit of a taste of the many different ways in which individual differences are manifested in alcohol misuse. But I also want to make clear that this is not specific or unique to alcohol misuse. All of the neuropsychiatric phenotypes, traits, disorders that everyone in the audience and online is interested in involve the human brain and human behavior, and these are, by nature, extremely complex. So I hope this inspires you to also consider these complexities in your own research and study designs.
Yeah, so first of all, Jeanne, I think you were by far too modest saying, talking about your role in neuroscience, because you showed us really beautifully how you can dissect using genetic tools the different components that are underlying such a complex trait as alcohol misuse, right? So we can learn a lot, I think. And it is also an entry point, like you said, into neuroscience, in trying to find the right genes for the right components and the right aspects of behavior. So I think it's a great contribution, but let's see what the audience has in terms of questions. Okay, so let's take a question. Do you look at epigenetics, knowing that alcohol influences expression of genes via epigenetics? Yeah, sure, that's an interesting question, right?
Yeah, absolutely. I don't personally look at epigenetics, but I think it's a super interesting role, both the epigenetics of other influences, environmental context, how that modifies alcohol use outcomes, and how alcohol use modifies your epigenome as well. I think those are super interesting, and I wish I knew more about them. So if anyone else, hopefully someone else in this audience can answer that question, and we can connect the question asker and the possible answer in this room.
Right, okay. Yeah, and another interesting question, of course, is the people that you involve in these studies, they now come from the UKB, the UK Biobank. And the question really is, are the alcohol use problems really good represented in such a group, right? Because that's, of course, the input for your studies. So can you comment on that?
Yeah, so the UK Biobank is a large population-based study. It certainly has a healthy volunteer bias, so people who join are more likely than average to have better mental and physical health. But alcohol problems are extremely prevalent in most societies, definitely in the UK. So it's a subset, as alcohol use is intertwined with most societies, most cultures. So it's actually pretty easy to study in many contexts. Right. And if you have if you're starting with 500,000 people, which the UK Biobank does, even if you have to subset down to a smaller portion, you still have a pretty substantial number that have serious problems, and an even larger number that have moderate problems at some point in their life.
Right. Maybe your second factor, your beer factor, came from the UK then, maybe specifically. Yeah? You never know. Yeah, we'll find out. Okay. And there is a question, of course, regarding environmental factors, as you can see. How did COVID or lockdown period may have been impacting on alcohol use?
Yeah.
So what do you think?
Ooh, that's an interesting question.
That's a tough question.
One. I think obviously it has to be measured very well. In the other study that I mentioned, Spit for Science, they did launch a survey during the COVID lockdowns to specifically assess mental health and alcohol use, drinking behaviors. So that's one of the samples that I want to use for following up some of these research studies. So that's a really good point that I can maybe look at and answer in another year or two. Right.
Maybe a last one. Clinical implications. How do you think about them?
Yeah.
Bridging.
Yeah.
Bridging to clinic.
Absolutely. Yeah. So I think if we're already breaking down the genetic influences on something complex like alcohol misuse into the genetic influences on severe alcohol use or binge drinking specifically, or in this group of people with this environmental exposure, we're already getting some clinical insight into what are the group of people, the group of patients best served by this genetic risk score versus that genetic risk score, or that biological mechanism that's implicated in one versus another. So I think we need to have some more actual experiments and some more implementation of these results to be able to speak more clearly to the direct clinical implications. But I think just by looking at these different dimensions, different subgroups, we already get a lot of insight into what are going to be the most important people or types of alcohol misuse or whatever other neuropsychiatric phenotype you're looking at. What is going to be the most tailored place to bring that genetic information into account?
Yeah, okay, Jeanne. Jeanne is in the complex trait genetics group of Daniela Postema. If you want to talk to her, you will find her. She has done great research also on Alzheimer's and intelligence, and there are GWASs that you worked on. There's a lot to learn from all these studies, so we look forward to the future. Jeanne, thanks a lot for today.
All right. Further to Jeanne's apologetic statement that she was just a geneticist and apologizing herself for that. Actually, I'd like to underscore that we had a neurologist on stage, we had several neuroscientists on stage, a psychologist / neuroinformatics, a biophysicist, and just a director. Okay, so don't be apologetic, we need all, and thanks for the great lineup speeches, also to Mototaka, to Jennifer at home, I hope she is online, and to Betty. All right. What are we going to do? We're going to have a break. So for your audience at home, we're going to be back in 30 minutes, 11:45 sharp, for the Story Slam. That's going to be led by Priyanka. Enjoy your coffee break. Mix, mingle, enjoy. Thank you.